Hello everyone, today we are going to see another very exciting system in the light microscope segment, and this is again from the Zeiss. This is first in India and this is none other less the Lattice Lightsheet 7. So Lattice Lightsheet is a very pretty new technology which is an addition to the Lightsheet system which Zeiss had already launched in India, and this is the first installation in India. So let's have a look at the system. As you can see, this looks a very sleek and compact design. Yet another, you can see the white and black design, which is more or less a trademark of Zeiss designs, and looks a very small footprint, compactly designed, where you have the sample holder on top, and in this sample holder you have on stage incubation system. So this is temperature controlled and closed incubation system, which you can see. This is the glass on which the sample can be viewed from top, and this is where you see the sample holder in the form of your lab take chambers, in the form of glass slides can be put here. Also small petri plates can also be accommodated. You have the sensors. These sensors will make sure the laser will only fire when this is a closed system, so that you do not do mistake while putting your live cell setup. Once this is closed, you have an extra lid and an overview camera on top to give you the full idea. So more or less this is a futuristic design, and in this design you get an overview camera just to see whether your sample is aligned properly within the incubation box. And once you close this, you are good to start with the imaging. You have a LED light here which indicates whether the lid is completely closed. The moment I take it out, the LED goes off, and once the lid is completely positioned in the correct position and ready for imaging, this LED will turn on. In addition to the Lattice Lightsheet 7, the nomenclature, as you can see here, it is written right in the front with a clear contrast, and you have the Hamamatsu Orca Fusion camera. So this is a high quantum efficiency camera for low light signals. For signal weak is very less, you have a quantum efficiency more than 90%, and also not to forget the frame rate, means for signals which have a poor SNR, for sample where the dynamics are involved, you need to do fast imaging for your volumetric sample as well as for your thin samples, maybe monolayer cell line which you are using it. This gives a wonderful microscopic images as a output. So next you have the same laser module, which is the recent laser module. This is, as I already showed, in the SIM 5 system. This is the laser module, a very compact design, yet again compact, sleek, but a very powerful. As I already mentioned in my SIM 5 video, that this is a laser 2.0 version, means the same box which LSM 900 had the laser, the similar type of box size, but now it is more powerful. So you can do with minimal laser exposure, you can get a considerable amount of signal emission from your sample. And the design of all huge electronics has been made as miniaturized as possible. It is a very minimalistic design from Zeiss with live cell setup. This is again from IBD. So this IBD is working closely in association with Zeiss, and you have this which controls the temperature, the humidity and the CO2 to maintain the incubations inside the sample holder. Let me demonstrate how easy to use this for your research. It simply starts by placing your sample in the incubation chamber and focus your sample in the bright field mode. Locate your sample, adjust the focus. In the next step, we switch to fluorescence mode, see in the live view, and start Lattice Lightsheet imaging. The system performs the full volume scan, as you can see here, and calculates the projection of the entire volume within seconds.
Imagine if the speed and gentleness of light sheet microscopy could combine with the high resolution of confocal microscopy. Here it is, readily accessible for your live cell imaging. Access this advanced imaging technique with your standard cell culture dishes and cover slips. Simply place your sample in the stage-top incubator to start your experiment. The unique auto-alignment procedure eliminates any manual adjustments. Gentle transmission illumination ensures that your sample is quickly located. The oblique detection provides natural contrast, giving you an overview in seconds. The 5-axis stage levels your sample with the highest precision and speed, compensating for even the smallest deviations in carrier dimensions or sample position. Ingenious beam shaping technology now prepares the light sheet for high-resolution imaging. Once inside the system, the laser beam is flattened and expanded to a sheet, then separated into its constituent colors en route to the spatial light modulator, which creates lattice structures according to the sample requirements. Finally, the structured beam is dithered to create a smooth light sheet before it enters the core optics. The monolithic design of the optical module ensures robust and alignment-free imaging. The light sheet illuminates your sample plane by plane, performing up to 3 volume scans per second with near isotropic resolution. Refractive index changes between cell culture media, cover slip glass, water immersion, and air are automatically corrected by unrivaled Zeiss optics. After passing the detection objective, the fluorescent light can be additionally filtered to your requirements. The entire procedure, from placing the sample to the first image appearing on your screen, takes less than a minute, so you can spend more time on your experiments. A controlled incubation environment and the integrated auto-immersion system enables unattended experiments over hours and even days. Zeiss Lattice Lightsheet 7 gives you high-resolution, long-term 3D imaging with next to no sample damage or photo bleaching.
So this is the Lattice Lightsheet 7 in a very comprehensive manner. If you want to check out the further details at depth, please check out the link below and explore more. Thank you.